The aim of Research Area 3 is to study inter-organ communication and responses to environment during aging and in aging-related diseases.
For example, we're very much interested in understanding how diet affects the aging process, how interaction between microbes in the environment affect aging and homeostasis, including aspects like inflammation and health in general.
We would like to understand how hypothalamus manages to mediate the complex regulations and how these regulations change during aging, because these changes, they do lead to age-related diseases. Whether we're hungry or not, whether we feel cold or warm, whether we're in danger, whether we're stressed. Hypothalamus integrates this whole information and then has to transmit a decision about about the major need of our organism. And so when you think about it, it is really surprising that we managed to keep these regulations like more or less in order for so many years, right? Because it's so complex. And if something goes there out of the equilibrium, then you can imagine that the whole system will be disbalanced. There are many different cell types in the hypothalamus which express different neurotransmitters, so they are able to excite or inhibit other neurons. And that's how, for example, food intake will be regulated, and changes of food intake, they lead to obesity and then also to potentially to diabetes.
This interaction between the organism and the external environment may be affected during the aging process. So, for example, one of our main hypotheses in my research group, for instance, is that the interaction between the host immune system and the external environment, including the microbes, it's really key to maintain health. And when the immune system starts aging, for example, when there is this phenomenon called as immunosenescence, which is associated with inflammation and what some define inflammaging, which is a very interesting term. So what happens is that also the microbial environment starts changing, and that in turn will affect the equilibrium, the balance, and the health of the organism itself, and so affecting it, therefore aging.
The great thing about the Cluster of Excellence that we are able to work across different scales, different methods, and in many different model organisms. And our methods, that's why they range from a single cell level or single gene level or subcompartments of a cell to living organisms, so we're able to change, to record and to change activity of a given cell type in a behaving animal, in a behaving mouse, and study the effects of a particular cell group on behavior, on dynamics of brain regions, and also on bodily responses.
Killifish is really the main model system that we use in my research group. So these are spectacularly short-lived organisms. They just live about 4 months, and their name is turquoise killifish, and they come from Southeast Africa, and we study them because they are short-lived, and so we can really perform many tests on killifish in a relatively short amount of time and know the results very quickly. They age just like we do, so they undergo like a whole range of age-related dysfunctions. So, for example, they undergo cognitive impairment, so they are less able to learn new tasks as they get older. They become less active. They also become less fertile with age. They develop cancer and many other features that are also associated with aging in humans. So they recapitulate in a way, in a short amount of time, many of the features that matter for aging also in humans. So we can use them as a system to study aging in an accelerated in a short time. We studied these fish in their natural habitat. So we collect fish, we study their life cycle in nature, and also their microbes associated with them in their guts. And we study aging in the field, in nature, in this species. So we do this research both in the laboratory, in this very artificial environment, if you want, but we are also very much interested in how aging affects fitness in the wild in this fish. What we did was to deplete old fish from their resident gut microbes using antibiotics. So we put antibiotics in the water where the fish lived overnight, and so there were no many bacteria left in the intestines of this fish. And then we took microbes from the intestines of the young fish, and we exposed them to the fish treated with antibiotics. And those fish treated with antibiotics, now exposed to the microbes from young fish, were ingesting them. They were literally eating them. And then so this was working, this happened for 12 hours. And then what we did was to test whether the transferred microbes indeed settled in the intestines of the recipient fish. And this is exactly what happened. And the individuals receiving microbes from the young donors were actually living longer than those that were receiving microbes from the same age donors. So we also had a control groups where middle-aged fish were receiving microbes from the same age fish, and so those guys receiving microbes from the same age fish were not living longer. So we had a direct impact of the microbiota from young fish to the health of the middle-aged fish. Our finding so far is that environment matters in modulating lifespan. This is already well established in the aging field. We know that one of the most effective interventions to modulate lifespan and aging in model organisms actually is dietary restriction. So this is already well known. Now we're trying to understand the mechanisms that connect those facts, right? So we want to understand the molecular basis behind this intricate modulation of lifespan and aging by the environment.
It is known that occurrence of sleep disorders is higher in older patients. So the older we get, the higher is the probability that we develop sleep disorders. And for now, it is not known which changes in the hypothalamus lead to the sleep disorders, but what is known that activity of cells in the hypothalamus mediates particular sleep stages and also wakefulness. And now we would like to understand which age-associated changes in the hypothalamic circuits mediate the sleep disorders.
For a long time, immunologists have thought about microbes as our enemies, and the immune system has been thought as something that protects us from invasions from pathogens and parasites. But there is really raising an evidence that function of those microbes that has to do with beneficial effects on hosts. And so if we can understand better how these interactions have come about, both evolutionary but also mechanistically, we can probably also harness this understanding, this knowledge to even engineer probably microbes to provide us beneficial molecules and factors that can help us fight off or prevent in those that now we consider inevitable age-related diseases, for instance.
When you think about aging, of course, the first think about age. The first what you will be thinking about as you think about age-related diseases will be, let's say, Parkinson's disease or Alzheimer's disease, and of course they're devastating diseases. But actually, aging starts much earlier than neurodegenerative diseases can occur, and there's dysregulations which start pretty early, actually, in lab before we. So at this time, hopefully, we think we are not too old yet, but the changes they already start, and they lead then to all the devastating diseases. And of course, age-related diseases are not only neurodegenerative diseases in the sense how we think about them, but also, for example, diabetes or obesity, and they're also widely spread, and they're very dangerous because they're associated also with cancer, with heart diseases, so they affect the whole body.